Okay, so now I want to do the histology of the esophagus. We want to do an in-depth look now. So the histology of the esophagus is the same as the histology of the rest of the GIT, but however there are a few marked differences, right? So to begin with, we have the epithelium. Esophageal epithelium is stratified squamous epithelium, stratified squamous non-keratinized epithelium, because this one has a protective action against the abrasive nature of the food bolus as it passes down the esophagus. Okay. Then for the glands, we have esophageal glands proper, which lie in the submucosa, and we have the second type of glands, which also called esophageal cardiac glands, okay, in the lamina propria. So the difference between these glands is that esophageal glands proper here, they secrete gas to offer protective action and also lubrication of the bolus as it passes down the esophagus. But the esophageal cardiac glands here, which lie in the lamina propria in the terminal portion of the esophagus, right? These ones will offer protection. In fact, they will neutralize the contents in the event of gastroesophageal reflux. They will neutralize those contents. Okay. So for the muscularis mucosa, we have the longitudinal layer being thickened in the proximal part of the esophagus so as to aid in the swallowing. Okay. And then for the muscularis externa, yeah, the muscle in the upper third is actually striated, right? Purely striated, right? Then the middle third we have mixed striated and smooth muscle fibers, and then in the lower third we have purely smooth muscle fibers. Okay, then we have the serosa. Remember serosa, we said it covers intraperitoneal portions, so the abdominal esophagus being intraperitoneal is covered with serosa, right? Is covered with loose connective tissue and mesothelium. Okay, then the thoracic esophagus here, the thoracic esophagus is covered with adventitia, right? So that it can be firmly attached to the posterior thoracic wall, right? Okay. Alright, so this is a picture micrograph of the esophagus. We can actually see here the esophagus presenting a branched appearance in its normal collapsed state, right? And then we can actually see this stratified squamous and keratinized epithelium. We can see the submucosa here with mucus glands in the submucosa and then lymphatic nodules in the submucosa. Okay, then the muscularis externa is visible here. We have the inner circular layer, this one, and then we also have the outer longitudinal layer. Then for the covering, in this time we have adventitia, right? So the fact that we have adventitia tells us that this section was taken from the thoracic part of the esophagus. Okay, so now let's look closer at the esophageal mucosa. We have this epithelium, stratified squamous. We have our lamina propria, which is loose connective tissue with diffuse lymphatic tissue, blood vessels, sometimes glands. Okay, we have our muscularis mucosa. Yeah. Okay, then here we have our submucosa with this gland, which is the esophageal gland proper. Then this is the duct of this gland. Okay, right. In the submucosa, remember, also find the larger blood vessels and lymph vessels. Okay, then here is our muscularis externa. Okay, let's look at the gastroesophageal junction. Here we have a transition from our stratified squamous non-keratinized epithelium to a single columnar epithelium here. Okay, so let's go to a more visible picture. Right, stratified squamous epithelium to columnar epithelium. So for the gastroesophageal junction, we actually see that we have a transition here. Okay, we have a transition from stratified squamous epithelia, 2 simple columnar epithelium. Okay, so that's notable here. Also have esophageal cardiac glands here. Okay, then also have gastric cardiac glands. So for the gastric cardiac glands, we look them in depth when actually do the histology of the stomach. Okay, now let's go to the histology of the stomach. Okay, so the general structure remains the same with a few differences, and we are going to talk about here. So to start with, generally in the fasting state, okay, when there's no food in the stomach, the lumen of the stomach presents with mucosal folds, which you actually call rugae. Okay, but these ones, they are temporary. They allow the stomach to distend when food enters the stomach. Okay, so it can distend without mucosal injury. All right, for the epithelium, it's simple columnar. Okay, we also have glands in the mucosa of the stomach. Right? We do not have glands in the submucosa, but we have glands in the mucosa of the stomach. Okay, these glands, they will be extending from the muscularis mucosa, right, through the lamina propria, and they will be opening into the lumen via what you call gastric pits or foveolae. Okay, right. So another notable difference for the stomach, we have for the muscularis externa, we have those 2 layers, the inner circular and the outer longitudinal. We also have a third layer, all right, which you actually call the inner the oblique layer. This is the innermost layer, right? So it is obliquely oriented. Okay, then we have the circular and the longitudinal. All right, the stomach is covered by serosa because it is intraperitoneal. Okay, so this is the gross anatomical structure of the stomach. Here we see these folds here. That's actually what you call rugae, and these are temporary. Once food enters the stomach, it will distend and they will disappear. Okay, alright, so this is the region of the cardia. That's where we find the gastric cardiac glands. Here we find the esophageal cardiac glands, and this is the gastroesophageal sphincter or the lower esophageal sphincter. Okay, this is the region of the fundus here. This one is the fundus, and then this one is the pylorus. This region is the pylorus. Okay, here we actually find another sphincter here, the pyloric sphincter. Okay, so the pyloric sphincter instead is formed by thickening of the circular layer of the muscularis externa, but this one is a physiological sphincter. Okay, so under normal circumstances, we say that the fundus is mainly filled with air or gas. All right, so in the fundus and in the body, we will actually find what you call gastric glands. Okay, we'll look at them in depth later. Okay, so this is the general structure of the stomach hole. Right, we can see here we have gastric pits here. Okay, this is a gastric pit here, and deep within the gastric pit we actually form the gastric glands here, which are made up of different types of cells. Okay, so these ones, we say they are found in the mucosa, and then they arise from the muscularis mucosa up through the lamina propria and open into the lumen via the gastric pits. Okay, so here we have the lamina propria here with its blood vessels, lymphatic vessels, capillaries. All right, here our muscularis mucosa. Here we'll find our submucosa with the larger blood vessels, the larger lymph vessels. Alright. Here, this one now, the oblique layer of the muscularis externa. So this is the only part of the GIT where we find the muscularis externa consisting of 3 layers. This is the only part. Okay. Right. In between the circular layer and the longitudinal layer, we find the mesenteric plexus, right, or orbital plexus, which innervates the maxillaris externa. Okay, then externally we find the serosa, which contains the larger blood vessels and lymph vessels, right, which will further branch to give us these small branches. For the cardiac glands, remember we said we have the cardia, which is around the region or the proximal part of the stomach, around the region of the gastroesophageal junction. All right, so in the cardia we find glands which we refer to as the cardiac glands. Okay, so cardiac glands are normally they are tubular, twisted, and branched. So these ones are what we refer to as our cardiac glands: tubular, twisted, and branched. And then they open into the gastric pits, right, to the lumen. Okay, so our cardiac glands, they have they're made up of a few cells as compared to other parts of the stomach. Okay, here we mainly have our mucus secreting cells, our enteroendocrine cells. Remember, the enteroendocrine cells are those cells which secrete gastric hormones, right? Most of the gastric hormones will act in an endocrine fashion, which means they will enter into the blood, all right, via the portal circulation. Or they can also act in a paracrine fashion, where they will diffuse into neighboring cells and initiate cellular responses there. Okay, all right. So for the gastric glands, these are glands of the body and the fundus of the stomach. Okay, these ones are mainly tubular and branched. You can see here, this is our gastric gland here. These are mainly tubular and branched. Okay, you can see this one here. So these ones are the cells which we find in the gastric gland. Okay, this is where we find most of the cells. Okay, so basally we will find our chief cells here. These are the most basally located, the chief cells, and they are also basophilic. All right, we have our enteroendocrine cells here. These ones are what you call enteroendocrine cells. We have our eosinophilic parietal cells, these ones. Okay, so these are eosinophilic parietal cells. We'll explain why they're eosinophilic and why those are basophilic in a short while. All right, we also have stem cells in the region of the isthmus, right, around this region. That's where we have our stem cells, which will migrate upwards to renew the surface mucus cells, or they can run downwards to renew all the other cells which you find here. All right, then we have in the region of the neck here, we have what you call mucus neck cells. Okay, mucus neck cells are simply mucus secreting cells, but however the mucus they secrete is different from the mucus secreted by the surface mucus cells, in that the mucus secreted by the mucus neck cells is slightly it's less alkaline compared to that of surface mucus cells, and the mucus secreted by the mucus neck cells is also soluble. Okay, and it's less staining with with PAS, periodic acid shift. Okay, so we can see here in the region of the neck this lighter staining. You can see these lighter staining cells here. These are the mucus neck cells, and then this deeper staining, this is what you call the surface mucus cells. All right, surface mucus cells, these ones. All right, so remember, this is not the usual H&E staining, but this is PAS, periodic acid shift staining. Okay, right. For the pyloric glands, these are found in the region of the pylorus. Okay, so they are tubular and they are coiled. All right, you can see here, these are our pyloric glands here. they're tubular and they're coiled, and they have their cells, again, they are few as compared to those of the gastric glands. Okay, mainly have mucus secreting cells. We have endocrine cells interspaced within the mucus secreting cells, and we have occasional parietal cells. Right. So remember, in the region of the in cardiac glands, we do not find parietal cells. All right, but in pyloric glands, we find occasional parietal cells. They are not that many; they're just here and there. All right. So that's your pyloric glands. Okay, now this is just simply a structure, the structure of a goblet cell. We have apically here, we have our mucinogen granules. Okay, this is macrovilli, and then this is the nucleus. So the nucleus of a goblet cell is basally located, and then the granules are apically located. All right. Then this is the structure of a chief cell and the structure of a parietal cell. Remember, the chief cells are the secrete pepsinogen, okay? The secrete gastric lipase, okay? And then this parietal cell, it secretes hydrochloric acid, all right, and also intrinsic factor. All right, so the chief cell is mainly basophilic because remember, we have synthesis of these zymogens. All right, so synthesis of these zymogens requires abundant rough endoplasmic reticulum. All right, so you are going to find that in the basally we have loss and loss of rough endoplasmic reticulum. All right, and apically we have many zymogen granules. So this basally located rough endoplasmic reticulum will actually cause the basal portion of the chief cell to be deeply basophilic. Okay, but the apical will be slightly eosinophilic due to these granules. But however, in sections, the chief cells are mainly identifiable by their basophilic nature. Alright, so this is the parietal cell. The parietal cell is mainly eosinophilic in histological sections, okay, due to the abundance of mitochondria here. Mitochondria, we need them for transport. Remember, we have our pumps which will pump protons into these intracellular canaliculi. Right, the intracellular canaliculus. So the mitochondria here present this with eosinophilic presentation. Okay, and then for the pyloduodenal junction, this one here, we have the circular layer of the muscularis externa thickening to form the pyloric sphincter. Okay, so here you see this thickening of the inner layer, okay, to form our pyloric sphincter. Again, this is our pyloric sphincter, our muscularis pyloric sphincter made by the inner circular layer of the muscularis externa is visible here. Look, the longitudinal layer take no part in forming of this pyloric sphincter. Okay, right, so now we'll go to the histology of the small intestines. So this one resembles the rest of the GIT histologically, except a few differences. The epithelium is simple-columnar and is absorptive, okay, with microvilli. The glands, we have glands in the mucosa and glands in the submucosa, but most of the glands in the small intestines is in the stomach. We find them in the mucosa. Okay. We also have luminal folds in the small intestines, which we refer to as the plica secularis. Alright, we'll feel this later. We also have villi, which are folds of the mucosa, and with a lamina propria core. Okay, of the submucosa, where we find the fused lymphatic tissue and lymphatic nodules, and we also find lymphatic nodule aggregates in the region of the ileum. Okay, right. Then, for most of the part of the small intestine, they are covered by serosa because they are intraperitoneal. Okay, But however, the only portion that's covered by adventitia is the duodenum, excluding its proximal part, which you actually call the duodenal cap. Okay, so most of the duodenum is retroperitoneal, so it's covered by adventitia, and the remaining parts of the small intestines are intraperitoneal, covered by serosa. All right, so this is the gross anatomical structure of the small intestines. You can see these luminal folds. Right, these are what we refer to as the plicae secularis. All right, these are permanent folds. They are different from those folds which you find in the esophagus and in the stomach. They are temporal folds, but these plicae secularis are permanent folds. Okay, so this is our duodenum. In the duodenum, that's where we will find submucosal glands in all the intestines. In fact, from the stomach, okay, going on, we only find glands in the submucosa in the proximal part of the duodenum. Okay, and these we call them Brunner's glands. Okay. So the function of the Brunner's glands is to secrete alkaline mucus, which will neutralize the acidic chyme as it passes through the pyloroduodenal sphincter into the duodenum. Okay, so they secrete alkaline mucus to neutralize those acidic contents of the stomach as they pass into the duodenum. All right. So remember we said that we have luminal folds in the small intestines, right? We have folds, the plicae secularis, and also have folds on the plicae secularis. These are mucosal folds which will form what you call the villus. So this is a villus with a lamina propria core and a central lacteal in the center. Remember, we once said we can find smooth muscle fibers extending from the muscularis mucosa into the lamina propria. Right, these ones, smooth muscle fibers. Right, and they will facilitate with the localized motion of the villi themselves. All right. Okay, so but basically for the small intestinal glands, we can also refer to them as the crypts of Lieberkühn. Okay, these are located in the mucosa of the small intestines, and they are largely made up of mucous goblet cells here. All right, we have the Paneth cells basally. So these Paneth cells are located basally, mainly in the lower third of the intestinal glands, and the function of the Paneth cells, right, we say it's to secrete defensin, lysozyme, and phospholipase A2. So since it produces defensins, it sort of like acts in aiding innate immunity against invading microorganisms. Okay, right. So again, here you can see the Paneth cells. These are located basally in the intestinal glands. These ones, they're eosinophilic, right? That's how we can distinguish them. And they secrete lysozyme, phospholipase A2, and defensin, aiding in innate immunity. All right. So now let's look at the different portions of the small intestines. First of all, we have the duodenum. All right. The duodenum is the proximal part of the small intestines. Here we can see our villi. Alright, these are villi in the duodenum. Alright, the plicae secularis, they are not so well established in the duodenum, alright, than in the jejunum. Alright, so actually see their establishment in the jejunum. So for the duodenum, we can distinguish it from the rest of the intestines by presence of what you call the Brunner's glands in the submucosa. So remember, I said this is the only time we find glands in the submucosa, Brunner's glands. All right. For the jejunum, right, we have well established plica secularis, right, and villi present as finger-like projections. So this is our plica secularis. Another one here, plica secularis luminal folds. All right, then on the plica secularis we find our villi. Then on the villi we find our columnar cells, which have absorptive capacity, with macrovilli on their apical parts. Okay, so this is the ileum, right? It's distinguished from the rest of the intestines by presence of what you call the Peyer's patches. These ones, the assembly lymphoid nodule aggregates, okay, located in the submucosa. All right, on the anti-mesenteric side of the ileum. So by anti-mesenteric simply means the side which is opposite the attachment of the mesentery. All right. So now to conclude with, let's now look at the histology of the large intestines. So these ones have the same general structure as the rest of the GIT, right? With marked differences in the epithelium. The epithelium is simple columnar absorptive. All right. We have goblet cells. So remember, goblet cells are simply mucus producing cells, right? And the number of goblet cells increases down the digestive tract. All right. So we have few in the small intestines, and in the large intestines we have more and more of the goblet cells, right? In the large intestines we do not have villi, but however we have macrovilli on top of the columnar absorptive cells. All right. We also have glands in the intestines, and these glands are in a similar fashion to the glands in the small intestines. They are simple tubular, right? They are found in the mucosa. Okay, remember we do not have glands in the submucosa in the large intestine. Right. For the muscularis externa, the inner circular layer, right, we have an outer longitudinal layer, but the outer longitudinal layer now is organized into three bands, which we actually refer to as the tenia coli. All right. The serosa is present in intraperitoneal portions of the large intestines. For example, the appendix of the transverse colon, the sigmoid colon. All right. The serosa, actually, when you look at the large intestines, the serosa it displays numerous fat-filled pouches, which we refer to as the appendices epiplacae. Okay, those appendices epiplacae. Then adventitia is present in ascending and descending colon. So here we say that the serosa displays fat-filled pouches, which referred to as the appendices epiplacae. Appendices epiplacae, these ones, all right. Fat-filled pouches, appendices epiplacae, right. Then this one here, gross anatomically, the tenia coli, right. The longitudinal bands of the muscularis externa. Then gross anatomically, the large intestine is organized into sort of like circulations, which we refer to as the haustra coli. All right, so you have tenia coli, haustra coli. Okay, so these are the intestinal glands, which we refer to here. Intestinal glands, we find mucus secreting cells. Okay, we also find enteroendocrine cells, but we do not have Paneth cells here. Okay, so we say that the outer layer or the longitudinal layer of the large intestine is organized into three bands, which we refer to as the tenia coli. So here is one of the tenia coli. All right, here. So this is the inner circular layer, and this is the outer longitudinal layer here. All right. So now let's divert our attention to the appendix. All right. So as for the appendix, the histology is the same as the large intestine, but however we have aggregations of lymphoid tissue here. So lymphoid tissue is found, we find it in the terminal portion of the small intestine, forming aggregates. All right, so these are there to provide a defense against the large populations of bacteria in the large intestines. So remember, in the large intestines we have large populations of bacteria. So to prevent invasion by that bacteria, we find large masses of lymphoid tissue being present in the ileum and in the large intestines. So this is the appendix. We have our lymphoid tissue here. All right. These aggregations of lymphoid nodules, which you'll find here. Okay. As for the clinicals, we have GERD, gastric and duodenal ulcers, celiac disease, and the Barrett esophagus, and many more which I did not mention here. So for the GERD, this is gastroesophageal reflux disease, where we have gastroesophageal reflux. All right, reflux of gastric contents into the esophagus. All right, this will cause mucosal irritation, can result in what you call esophagitis, which is inflammation of the esophagus. Then we have gastric and duodenal ulcers, right? Gastric ulcers, these are ulcers in the stomach, duodenal ulcers in the proximal part of the duodenum. All right, these are mainly due to bacteria, number one, our Helicobacter pylori, which invades the mucosal lining, all right, and degrades that mucosal lining. Okay. We also have things like NSAIDs. Okay, they can irritate the mucosa and actually erode our mucosa, leading to the production of ulcers. Okay. Then we have celiac disease or sprue, celiac sprue. This is now more of a disorder in the small intestinal mucosa, where we have an autoimmune reaction against gluten, such that the villi will actually get damaged. So when we have damage to the villi, we have malabsorption. Okay. Also have Barrett's esophagus. This is where we have metaplasia of epithelium in the terminal portion of the esophagus due to progressive gastroesophageal reflux disease. Okay, so the epithelium in the lower portion of the esophagus will undergo metaplasia to become simple columnar, okay, is in the epithelium of the small intestine. So it's sort of like changes to become epithelium of the small intestines. All right, that's Barrett's esophagus. So that changing, that metaplasia can lead to the development of uncontrolled division. Okay, can lead to cancers, can lead to tumors and cancers. All right. So thank you. This is the first part of the histology. Watch out for the second part, where we'll be discussing the histology of associated digestive organs. Okay.